Howdy, my name's Emily Alice, and today I'm going to tell you how Apis mellifera processes the pheromone isopentyl acetate with the antennal lobe to determine when to sting. So, let's go over what a pheromone is, just as a refresher. A pheromone is a secreted or excreted chemical factor that triggers a social response in members of the same species. Social insects, in this case honeybees, employ several glands in order to biosynthesize their alarm pheromone. Alarm pheromones appear to be normal constituents of the Dufour's gland secretions. The contents of Dufour's gland are secreted in a mixture with the honeybee's venom, which is an excellent defensive adaptation since the alarm pheromone is generated whenever the venom is ejected. The mandibular glands of the honeybee also contain a chemical release of alarm behavior, which is, however, considerably less ethopotent than the chemical release from the sting. The pheromone that elicits the most stinging activity from the honeybees than any of the other defensive pheromones is the isopentyl acetate, also known as IPA. It acts as a target marking pheromone, guiding other defenders to the sting site. In the picture attached, a study done by Wang, Sato, Gurifa, and Zhang shows how 8 sting pheromone components evoke specific activity patterns within the antennal lobe of workers who didn't perform the stinging. Gray indicates intensities lower than 0.2%, red indicates intensities greater than 0.6%, while blue and orange indicate intermediate intensities. A gland within the honeybee that's worth noting, along with Dufour's, is the Nasonov gland. This gland in worker bees produces a pheromone, which is a blend of nerol, geraniol, E-N-Z-citrol, nerolic acid, geranic acid, and E-E-farnesol, that acts as an attracting signal. This pheromone is used for aggregation, which is swarming. In the attached picture, a study done by Trillin and Richard produces results showing that there are 8 to 10 alarm pheromones present when a honeybee stings. Why do worker honeybees sting? The answer is pretty obvious, but it protects the hive from whatever the threat is at that point in time. The guard response of the worker bee is the most important. Unfortunately, when a honeybee stings, it is typically fatal. When the worker bee feels threatened, it retracts the stinger from inside its abdomen. The stinger is dropped into place, and the worker then stings the threat. The barbs in the end of the stinger can get stuck within the skin layer of the threat, which breaks the sting away from the worker, disemboweling it. The worker later dies. When the sting drops down from inside the honeybee, the alarm pheromones are released to alert other worker bees within the colony. The pheromones secreted by the worker that had just stung come from the sheath glands within the stinger, which is where Dufour's gland is located. All honeybees have the same intricate head parts that allow them to communicate with pheromones and complete tasks. However, not all honeybees look the same or perform the same tasks. For this topic, we'll be focusing on the worker honeybee. These bees, as shown in the 2012 Encyclopedia Britannica picture, are noticeably smaller than the queen and the drones. The worker honeybee is always a non-reproductive female. Because of this, their ovipositors are used as stingers. Some of the different tasks a worker does includes the housekeeping, feeding the queen, collecting the pollen and nectar, making the wax, and most importantly, protecting the colony. The head of Apis mellifera has some of the most important parts that work to help it survive and carry out day-to-day tasks that are essential for their survival. However, the most important of those parts include the antennae and mouth parts. The antennae are a bee's source of data collection and contain receptors for touch, taste, and smell. Their antennae can also detect the temperature, humidity, and carbon dioxide in the air, as well as gravity and wind speed. Much of what a bee knows arrives through those two filaments. The mouthparts of a honeybee are the chewing and lapping type, which is perfect for collecting nectar and pollen. The honeybee can sense pheromones with these two parts, which is the reason why the antennae and mouthparts are so important. The pheromones are the way in which a honeybee communicates with one another. Now that we've discussed the outside of a bee head, let's discuss the inside. Their bee brains are so tiny, yet so complex. Listed here are the different components within the bee brain. However, the neural pill to be focused on is the beta lobe, which, in this picture on the right, the beta lobe is the red section right in the middle. This lobe is located dorsally within the bee brain and looks like 2 little golf balls, as you can see. It's a neuropil, which is a dense network of interwoven nerve fibers and their branches, as well as their synapses. It processes olfactory information, which is the sense of smell. In a study done by Wang, Sato, Gurfia, and Zhang, they found that sting preparation evokes glomerular patterns that are distinct from those of individual pheromone components. The sting pheromones processed in the worker honeybee's antennal lobe follows the same principles of general odors. The chemical structure of odorants is the main determinant of glomerular activation rather than the pheromonal value. As I mentioned previously, the beta lobe deals with any and all olfactory information the bee takes in with their antennae and mouth parts. Within this lobe, the antennal lobe is located. The antennal lobe is a sphere-shaped deuterocerebral neural pill in the brain that receives input from the olfactory sensory neurons in the antennae and mouth parts. Each neural pill here is composed of the arborizations of over 17 small interneurons in similar architecture called Kenyan cells. Kenyan cells are the intrinsic neurons of the mushroom body, which is a neural pill found in the brains of Apis mellifera. Golgi staining done by P.G. Mobbs for his research for the Philosophical Transactions of the Royal Society Journal demonstrates that these neurons can be divided into 5 groups distinguished on the basis of their dendritic specializations and geometry. It should also be noted that within these antennal lobes there are about 100 glomeruli each, which is a lot. Glomeruli are a cluster of nerve endings, spores, or small blood vessels, or in particular, a cluster of capillaries. This makes the antennal lobe sensitive to any and all information the honeybee takes in, which helps explain why they're fantastic at communicating with pheromones. Each olfactory sensory neuron in the antennal lobe expresses a single odorant receptor type and targets the same glomeruli as other olfactory sensory neurons. So, when honeybee senses the isopentyl acetate, projection neurons, which generally receive information from a single glomerulus, project to higher brain centers such as the mushroom body and the lateral horn. The interaction between the olfactory and receptor neurons and projection neurons reformats any information taken in by the antennae and mouth parts and creates a sort of code from the sensory neurons before the information from the pheromones are sent to higher brain centers. Once this is complete, honeybees from the same colony head in the direction of the pheromone released and know that they must fight off the threat of one of the other bees in the swarm had already attacked. As shown throughout the slides, Apis mellifera truly have an intense, intricate system in place with their antennal lobes for them to communicate with others. The secretion of the alarm pheromone isopentyl acetate is one of the many evolutionary traits that help them succeed and protect the colony. Without being able to sense these pheromones with their antennae and mouth parts, colonies would likely collapse and fail. Thanks for watching!